I would like to introduce tonight's speaker, who is going to be talking about a topic that I love, which is eating. But more specifically, he's going to be talking about how our brains react when we eat and how that can lead to addictive behaviors at the chemical level, I think. Yeah, at the chemical level, sort of.
Sure.
How that can be similar to other addictive behaviors like drugs and whatnot. And so without further ado, I'd like to introduce Associate Professor Matt Will from the Department of Psychology. And you.
Thank you. Okay, you can hear me, right? Right.
Unless you give them that
Yes.
chemical, right?
What was it? So I was just sent this article yesterday. I made a joke a couple weeks ago in a talk about, they asked me what I had shown with my data, and I said, oh, because I had found where I could inactivate this portion of the brain, and the rats would stop eating it, but they wouldn't stop from going to it. So they kept going to the food and spending time looking at it, basically hovering over it, but they stopped eating. So it's like I knocked out their desire to eat, but not their desire to approach it, and it looks like they want it, but they just wouldn't eat it. And I said, oh, maybe I'd found the circuitry of guilt. Like they have this guilt right at that last minute. But there was another research finding that came out recently that, regret is what it was. That they think they have a model of regret in a rat.
Like remorse or whatever.
Yeah. So, how much, but your question makes me realize that I haven't talked enough about conditioning and how much that goes into, and it's all this implicit conditioning, which is just conditioning that happens subconsciously that we're all maybe aware of, but advertisers are very aware of this. It's not subliminal. It's just out there, and one factoid I can give you that I just learned about this year is that, I don't know if you've ever thought of this, but of all the information coming in through our five senses, how much of it actually reaches our consciousness, our awareness, right? So you think about all the information coming from your senses, and this, of course, has to do with conditioning. You think sometimes, okay, I'm only conditioned to things that I am aware about, which is true. There's certainly conditioning happening on that level. But this underlying implicit level of conditioning that happens is the vast majority. And so the percentage of information coming through our five senses that reaches our consciousness is 0.000046%. So out of like 16 million bits of information per second, we actually take in to our conscious awareness about 60 bits. So it's a very small amount. And so yeah, there's not much thinking going on in most of our behaviors. And you maybe heard of, we had a guy come to speak on mindless eating, to where he takes advantage of that fact, where our feeding habits are governed by things we're never conscious about. And the one thing I remember from his research is the bottomless bowl of soup, where he would fill the soup bowl from underneath, and he would see how much soup these people would actually eat if they weren't aware of that. But they just have the soup continually, just I guess slowly going down. It has to go down a little bit. But another thing this guy did, I can't think of his name, but he would go into cafeterias and move food, and so normally the desserts are right next to the cash register. And so, of course, that's the last thing you see before you check out, and so you're going to grab some, more likely than if it was further away. So all he did was move the fruits and vegetables up near the cash register, move the desserts further away. Sure enough, kids start eating more fruit, eating less dessert. So it's all this subconscious processing that your brain does that governs how much you eat. And those variables, are in competition with the genetic predisposition, driving your motivation to eat. And so, it's hard to say which one has more power, but it's going to be different for each individual, right? So, good question.
Hi.
Hi.
I'm kind of curious as to how you're collecting data. So you're talking about food choice, and you're also talking about measuring brain activity. So I just have this image of you know, a little rat with dominoes and like a rice cake wearing a little metal mesh helmet.
Exactly. Yep.
That's what I think.
Yep. So yeah, some of the methods we use, you know, we have a lot of what I do is we operate on these rats, and we are able to put in a little tiny tube that we can later inject slide a needle in. So we do a lot of very small, we call them micro injections, where we can activate or inactivate a certain region of the brain. And we can, you know, choose our drug, so we can choose what receptor, or what peptide, what neurotransmitter we want to influence. And we do that while they're wide awake, sitting on our lap. So the endorphin experiment I talked about, where we actually add additional endorphins to their brain, we have the cannula that's already been placed in their brain a week before. They've recovered, and now they're sitting on our lap. We inject them, and they're sitting there, and they've gotten habituated or used to it, and so they're very calm. Take the needle out, and then we put them in their little feeding chamber, and they might have a choice between two diets, or we might put them in with the running wheel with two diets available or something like that. And then we look to see what their behavior, what they exhibit in behavioral, different behavioral standards over a couple hours or however long the drug lasts. So I don't know if that gives you a better image of what we do. And I'm sorry I didn't bring any photographs I can hand around, but PETA would be all over me.
So do rats have vitamin deficiencies like humans do?
In the laboratory?
Yeah. Well, or can you simulate that and then see if they go for a different type of food?
Yes. Actually, a colleague of mine just did a study with thiamine deficiency. So yeah, you can do all sorts of things. That's the nice thing about working with rats, right? You can, there's still these ethical guidelines you obviously follow, but you can do more invasive and potentially dangerous interventions. So now, did you have something specific about vitamin deficiencies that?
If they have one, do they crave the foods that will help alleviate that?
I don't know. That's a quick answer. So I have not done any research on that, but it sounds like you're familiar with something that you read about.
No, I'm really not. I mean,
No.
I've had different deficiencies in the past, but I don't find that I really crave those.
I can't see why not.
It seems like that would be natural.
Right. It's certainly not a question that would be off the table. Like, oh no, vitamins, nothing to do with food intake. So yeah, certainly that's something that, if it's not being studied, which I'm sure it is, but it should be studied. And you hear about vitamin D deficiencies quite a bit. We have a researcher on campus that studies a lot about vitamin D deficiencies and all sorts of symptoms, what that leads to. So whether they've looked at other vitamins that are more likely to cause changes in feeding behavior or make you more prone to food addiction. There's so many indirect ways that you can get to a place where you have addictive eating. I was like, what's the name of this talk? Addictive eating. So you can imagine a vitamin deficiency that would lead to make you more likely to become depressed. All right, well now you're depressed. Is there any research on the relationship between depression and eating? Yes. So there you go. The answer to your question, yes. Vitamin D deficiency causes eating disorders. But I'll have to email you the references. Yeah.
Do you have a theory on whether you should eat like fewer larger meals or a greater number of smaller meals like throughout the day?
Snacking, right. I bet this comes back to, I can't make a blanket statement like everyone should be snackers and, avoid the big meals. Because we all have our different tendencies and reactions to eating a big meal. So whether or not one has been more associated with, like if you did a model in rats where you gave them the same amount of calories, but one group got their calories in 2 big calorie sessions versus spread out over 8 feeding sessions, what would that do? I would think actually, so you can just go to the drug addiction literature, which is a nice thing actually, because we've been studying drug addiction for decades. Unfortunately, there hasn't been, a cure for that, or even really good drug therapeutics. But drug addiction research has given us a lot of information about how the brain responds to rewarding substances. And there's definitely parallels between, for me, it's morphine, heroin, are the drugs of abuse that match up with the food that I give my rats. That's what I've been studying, is how high-fat substances, high-fatty foods parallel the effects on the brain that morphine or heroin show. And so with any drug of abuse, if you give it in just big shots, you're going to make that rat have a bigger high and a bigger drop, a bigger withdrawal. So if you gave that same amount of drug in smaller shots, you would definitely not see as much withdrawal. And so, and that's, of course, one of the trends of a drug addict is that they tend to use more and more drug as they develop tolerance. And so, if you're a recreational drinker or a recreational drug user, by the way, the majority of drug users never become addicted. And that has to, it has something to say about the use patterns, but it also has something to say about your biological predisposition. But yeah, definitely the schedule, we call it the schedule of reinforcement, or the schedule of drug taking or food taking or food, feeding, I guess, would definitely have an effect. Now, we might not all have the time. There might be some practical issues there with snacking, but good question though. There are definitely rat models out there that have looked at that.
In what ways are humans and rats like a fair comparison or similar?
Where are you? Over there? Oh. Well, are human and rats the same? What?
Are like a fair comparison? Like how are they?
Right. Am I wasting my time?
No, like compared to like a different animal.
Yes. What's funny, so I always used to say rats are way smarter than mice. You guys probably don't care about that distinction. But that was big for us rodent researchers. It was something that we actually held this belief for about 100 years. This article just came out where they actually challenged that theory, and they showed that no, mice are just as smart as rats. You just have to adjust things and make them smaller. So for the comparison between humans and rats, how similar are they? They're a mammalian model, so the nervous system or any other system in the rat is going to be very, very similar. Now, how similar? Well, I'll say 99% similar. But the model obviously has been useful. It's been kind of the go-to model, and a lot of fruitful findings have come out of it. There have been recent, data on different studies on the immune system and how infections, as we become more and more aware of how infections and antibiotics are not doing their job. So a lot of studies are being done on the immune system and how well that's holding up in our present environment. And they've found one study in particular where it was like what worked in the mouse, completely opposite in the human. So there's definitely exceptions, but luckily there are few. So it's a good model. I don't want to work on rabbits or dogs or chimps. I just draw the line at rats, and I think my fellow researchers and myself will hopefully advance the field of science. But good question, and it's good to have to defend it once in a while, because there is new research coming in every day where these exceptions pop up.
Well, I have a question. So high caloric or
addictive foods are usually a combination of high fat and sugar or high fat and salt. When you take the parts of the pieces apart, like the salt from the fat or the sugar from the fat, like the flavor enhancers from the fat or the flavor enhancers by themselves, are the foods as addictive when those components are separated out, or do you know?
No. I'm going to take a guess. I've been taking guesses all night. No. So, I'm just kidding. So the flavor enhancers, now could I answer that question in relationship to like artificial sweeteners, right? Non-caloric artificial sweeteners. Everyone thinks, great, I'm not getting any calories, but what does the brain think about these things like NutraSweet, right? So the brain, first of all, they're more potent, and the beverage industry they try to equalize it so that the Diet Coke is just as sweet as the regular Coke. So they do a good job of that. They obviously got tons of research on that they don't ever publish. Unfortunately, just like the tobacco industry, had in the past. So the question was, though, what does it do over the long term? And does your brain know the difference? Certainly your body knows the difference, like calories or no calories. But your brain is responding to the rewarding value of that. And if we're talking about drugs of abuse and making that comparison between certain foods like sugar and a drug of abuse, your body doesn't need cocaine. Your body doesn't need heroin. So what are we worried about, with, what happens when we use heroin or cocaine? It's what's happening in the brain that's what we're studying. So with these artificial sweeteners, that's what we should be concerned about, is what is going on in the brain that then could lead to changes in feeding habits. Certainly the Diet Coke itself has not added any calorie content to your body, but is it doing something to your brain that is similar to what sugar does to your brain? And then you think, well, what does sugar do to your brain that might be counteracted by the added calories in your body? So there could be something good about, getting the calories with what's going on in your brain. And so, of course, that was never studied. It's beginning, people are starting to study this. And so you might have seen a lot of articles come out on how, it actually has adverse effects. So you're worse off in the long term if you're drinking diet soda. So say no to diet soda. Get on the real sugar. Yeah, and with the artificial, I don't know what is artificial fat. Are they like non-caloric fat flavoring? That would be the same thing. Your brain wouldn't know the difference. You certainly, there is a difference because of the peripheral feedback. But when you taste it, and the reward of that is registered immediately, NutraSweet or an artificial fat, whatever that is, your brain's going to immediately register that as, oh, it's very rewarding. So it's going to be treating it just as it would in the sense of how it's changing those pathways in your brain that govern craving, later cravings, withdrawal, and relapse. So, yeah.
I was wondering, it seems like the rat and human body systems are similar, but a difference in them is intelligence. Like humans have, if they overeat, guilt or other emotions, and I was sort of wondering how you would compare that to a rat and what you think about these emotions involved in eating.
Yeah, so there's recent research, cutting edge research that shows they maybe have regret. So, but we'll have to replicate that before we can go. Put that on the front page. So, again, that actually brings up, something I've been thinking about a lot, because I used to be really interested in consciousness. And my friend and I had this website. It was called Neurophilosophy Café. So I emailed him this week. I said, I'm doing a science cafe. It's like, our website that never got any hits, and now I'm doing it for real. It's actually live. But we used to talk about a lot of things, and not necessarily related to our own science. But the idea of consciousness versus what's happening, in your body, in your brain, that's not ever filtering up to your consciousness, like I was talking about before. So there's very little that's getting up to your consciousness. So can you experience guilt subconsciously, without the, if we think about guilt or these emotions that we have, or any emotion associated with choosing to eat or not choosing to eat, you think of it on a conscious level, right? But it's certainly happening on a very non-conscious level. And that's where I think the similarities between rats and humans are very, very identical. We don't have, of course, the ability maybe is what's limiting us from understanding how conscious a rat is, and then to define the nature of that, whether they experience regret or guilt, or what that would mean for a rat. So we're limited with the tools. The fact they can't talk, they can talk actually, but we can't understand it yet. We're getting there. But they do give out these ultrasonic vocalizations, which, this is true, I'm not joking, but they communicate with each other, obviously, and it's being categorized and kind of broken down into a language by researchers. So that's kind of a new area of research. So maybe someday we can ask them, how do you think about this food? Do you want it? All right.
Okay, so at the beginning you said this kind of dichotomy between healthy and not healthy, the rice cake versus the high-fat, high-sugar food. But have there been any studies looking at one type of fat versus another and how that affects it, or one type of sugar? So say the fat from olive oil versus butter, or the sugar from fruit versus plain refined sugar, and how the different kinds might have a different response, or is it all the same response?
Right. I will defer to a nutritionist in the room. Do we have any nutritionists? So, yeah, it's interesting. Like when I began studying, what governs high fat intake in a rat, we chose it because it was just something that rats really love to eat. And, certainly you should know a little bit about, different types of fat and would it make a difference. And so that's just something I'm not very knowledgeable about. And so that's why I'm deferring to any nutritionists that might want to, give an answer. But it's a good question. I just don't have the answer for that one. You stumped me.
I have one.
Okay.
So when you look at these, getting back to the genetic versus non-genetic component, obviously there's a gray area in terms of genetic modification within one person's lifetime in terms of epigenetics, if you want to call it that. But so, do you look at the predisposition of a given rat in terms of its genetic line, its family, that type of thing, when you're taking the 1 kilometer rat in consideration versus the 20 kilometer rat?
Yeah, so that was Dr. Frank Booth, that he's developed this line of rats that run either very little or very a lot, and so he's been doing all these gene assays, looking at the expression profile of the genes, and so he's published a couple of papers looking at what is different as far as what polymorphisms, these different variants of different genes they would have that have been selected for, and then also how are they expressed differently. And so he's definitely found some differences, and so there's definitely a huge area of research on how this difference on the genetic level contributes to that running distance. And essentially, because they're being raised in the same environment, identical environments, his rats, can be said to be running different levels, different distances, due to the selective pressures of the breeding. And it's just more of a question of what has changed genetically and what do those genes regulate. And so he's definitely answering that. Now, with some of the research I've done, we look at how the expression profile of certain genes are changed by food. So you might have a baseline before you ever experience a drug, or before you ever experience a gallon of ice cream. But then once you've taken that drug or eaten that entire gallon of ice cream, you might have altered your gene profile, the expression level of that. So I did a rat study once where we looked at what would happen to the gene that codes for endorphins if we gave this rat all they could eat for 2 weeks was chocolate and sugar. Not necessarily at the top of my list of palatable things to eat, But we gave these rats chocolate and sugar. It's a high fatty substance that these rats could drink. So no chow, no regular rat chow. They had a good time. So they had 2 weeks, they drank this chocolate and sugar. And what it did was it down-regulated or made this gene less active. So it basically was producing less endorphins. And the same thing happens if you give rat morphine for 2 weeks. So if you inject a rat with morphine or you let him administer morphine for 2 weeks, this same gene down-regulates. It's just, a compensatory process. So you get all this, extra endorphins being released by the diet or the drug itself is an endorphin, an opiate, then the genes will respond by producing less of it. And so you have that compensatory response. The problem is when you take the food or drug away, what happens? Well, now you predict, okay, you've got the baseline level of expression being dropped. Now you take them away, you take them off that drug. They used to get, up to here when they would eat or when they would take the drug for the first time, but now they're starting from down here, so you've got much further to go, so they're going to feel much, crappier basically to begin with, so they're going to need more of the drug, more of the food to get back to where they used to get, or just to get to baseline.
This actually brings you back to the observation that she was asking. If you take the pieces apart and you start introducing things like artificial sweeteners, which at the beginning you would imagine that they have some sort of behavioral component that mimics sugar, whatever it's going to be. But then after a certain point of time, you would imagine that the levels of endorphin or gene expression would eventually drop, and we reintroduce sugar. What would happen in that case?
Yeah, that'd be interesting. So, if it was an equal amount of sugar that produced the same amount of reward, which I would operationally define as how much endorphin does it release. So if the NutraSweet dose that you were getting from your Diet Coke, and now your idea of introducing sugar, now taking someone that was on Diet Coke, putting them back onto Coke with real sugar, but the same amount of reward or endorphins getting released in the brain, but now having that the caloric being processed. That was your question, right? So how would that then alter maybe how the brain is? And that it's interesting how neuroscientists, obviously we focus on the nervous system, but a lot of us only focus neck up, and so we're really focusing on the brain. And then you'd think, okay, I'm a feeding researcher, I would really focus on the digestive system and how all these signals feed back to the brain. And some neuroscientists, of course, collaborate, and I've collaborated with Dr. Frank Booth. He studies the periphery, and he understands a lot of these systems in the periphery. But it's definitely a question I don't know the answer to, first of all. But it's a interesting experiment to do, and that's the one I don't know how much research has been done on that. But I would think that these people, and there is quite a few labs studying these artificial sweeteners, and are they good, are they bad, and are they is there no ill effects of them? But it seems more recently it's leaning towards the bad, and how much they know about the introduction of the sugar back to the system, is that how that would all end up, I don't know. But yeah, good question. I would say, do it slowly, right? It's like, and then get your sugar from multiple sources. Add a little bit to your coffee, and then just drink half the can of Coke. So you're getting the same amount of sugar, but 2 different sources, and kind of spread it out. And drink it in different environments, so you're kind of weakening the conditioning that's occurred.
But coffee's a whole different issue, isn't it?
Yeah, it's good for you. Yeah.
So it seems like with what you're talking about right now, with changes in expression, or for example, the drinking some Coke and then some coffee, it seems like someone could make a lot of money off of making a synthetic drug. So I guess what I'm thinking of is like nicotine gum. It kind of helps you wean off of nicotine. Wouldn't something similar be possible here? If you know the signal, if you know what the brain responds to, you could mimic it and then kind of take it off slow, which could maybe help people stop eating. So.
Yes. So it's interesting. I mean, the nicotine patch, everyone's heard of that. They've got patches for a lot of drugs. I had this girl I went to grad school with. She then went into law school afterwards. So now she's a patent lawyer, but she's working with a company developing a marijuana patch, essentially. And it's not to cure people from marijuana addiction. It's to give them the medicinal benefits of marijuana, but through a patch. So, the idea of getting opiates, if that's what you want to, say, oh, I'm addicted to ice cream and it's because I get this opiate rush. So yeah, and it would be, it makes sense, the idea of having your opiates delivered through a patch, so that you wouldn't have the cravings, and you might then, of course, not eat as much of that food that was causing the problem, but you're still getting that reward. It all makes sense. Does anyone think he won't make $1 million by doing this? Or more, lots more than $1 million. But, so yeah, it all makes sense, in principle. Doing a substitute reinforcer just through, and especially by having it on a patch, that removes all the context of the normal drug taking or all the environmental variables associated with food, and that's what makes food so much more, I think, of a problem when you're trying to changing drug use habits versus changing eating habits. Just think of the environmental variables that are associated with feeding, the number of variables that are associated with feeding compared to a drug. First of all, you need to eat, right? So you need to go to the fridge. You need to see the food. And so there's just a lot of the variables that are surrounding eating behavior versus drug use behavior really, I think, makes it more difficult to alter that behavior.
So how close are you to, or is anybody to getting you talked about kind of like an endorphin injection that you could give to the rats to make them not want the high-fat stuff and whatever. How close are you to getting that into like clinical trials for humans so that they can avoid
Humans?
food and avoid gaining weight and all of the other, diabetes and things like that, and instead get that brain sensor working,
right?
Is that something that's even close to being in?
Oh yes, so there already have been clinical trials, and they fail, as they usually do. So it goes back to the idea that these systems that govern your response to any rewarding event, there's not an individual peptide or neurotransmitter that responds to the rewarding nature of food, and then there's a different one for sex, or a different one for social bonding, all these other rewards, exercise, gambling, what else do people enjoy? Drinking. They all act on the same area in the brain. So when you go in with a drug that is, of course, going to be taken orally, that's the first problem, where you're really creating a challenge. So it's going to bind to all the receptors in your entire body that it's designed to bind to. And you really only want to hit one aspect of the brain or nervous system. But what happens is, so the clinical trials I'm thinking of were in relationship to basically an anti-weed pill. So anecdotally, marijuana causes a change in feeding behavior, if you weren't aware. It's referred to as the munchies. So the first grant I got from NIH was actually to study the munchies in my rats. Yep. So I was looking at how opioids and cannabinoids, so this endogenous chemical that is the natural marijuana, how they interact to produce feeding. And so at that time, there was a clinical trial going on where they had an antagonist, so a drug that would block the receptor where your natural marijuana molecules in your body would bind. And they actually found this out, and it was in a trial. It was in a clinical trial to study this drug on nicotine cravings. So I think it had some effect on reducing nicotine cravings, but they said, wow, people are reporting that they're eating less and having less food cravings, which shouldn't have been a surprise. But anyways, then I think a new trial started where they really focused in on the feeding, trying to curb appetite with this anti-weed drug. And what happened was side effects, right? It was just because of the fact that these systems are redundant for these different behaviors, these different mental states. So people had depressed mood because endogenous marijuana is not just making us high when we go running, it's making us feel good during our engagement of any rewarding activity. So it's, it's contributing to that. So if you block that, with a pill you take orally, it's like, yeah, that's, it'd be a miracle if it worked. But it does sometimes, which is weird. It actually does. But there's always, there's typically side effects. It's just how many, cost benefit. So, yeah. But I'm not very close to becoming a millionaire. No. Yep. Any other questions? You enjoying your food? I didn't ruin it for you?
One more question.
All right.
I have one more question. So once you take the addictive food away, how long does it take for the rats to get over their withdrawal from whatever it is you've been feeding them?
Right. So I have not done many studies. I've done one where I actually was attempting to measure withdrawal from Oreos in a rat model. So I had this grad student. She came from another institution where they were giving rats this huge menu of junk food. And she said she thought that the rats that were getting the Oreos, compared to all the other junk foods, they were the nastiest, meaning more aggressive about when they were getting the food. So she thought it's because they were going through more withdrawal and it was changing their brains more than the other junk foods. So that was one reason we picked Oreos. The other reason, it's just fun to do a study with Oreos. And, free Oreos. No. So the withdrawal effect that we found was we were just going to measure. We gave them, I think, 2 weeks of Oreo access, and then it was in an automated feeding chamber where they would be measured for how much they put their face in where the Oreos were. So hopper entries where the food was, the Oreos, and so that on the day after the 14 days of Oreo access, we measured would they go and seek out these Oreos, and I think we had another variable, might have been injecting opiates or something, but the measure of withdrawal that we had, which was going back to this hopper, was probably just a bad measure because the rats are smart and they're like, no Oreos. I'm not gonna, they'll check a few times, but then they'll stop going back. So basically the difference between the ones that were getting Oreos versus the rats that were getting something less palatable. Like they didn't show much difference in that seeking behavior. But there is research where people design the study a little bit better and are actually trying to get withdrawal. And Bart Hobel at Princeton, he's passed away, but he studied, he really was modeling addiction to food just like you would model a drug addiction, trying to get all these phases, acquisition, maintenance, extinction, relapse, and all this. And he did it with sugar, and he would get withdrawal behaviors. And what he would have to do, though, because obviously with drugs of abuse, they're much more potent. You could dose them down to where they would be similar to what sugar would do. But typically drug addiction studies and the doses that are similar to what a human would take, are more potent. That's why people use them. And what he found is that he'd have to do this naloxone precipitated withdrawal, which he basically is injecting a drug that blocks opiate receptors. So the sugar, he believed, and he could actually measure, the sugar was releasing endorphins, just like I see in my research. And if you do that, he gave the rats access to sugar for a couple weeks, and then he would give them this blocker, which basically, if there was some withdrawal there, he's just going to exaggerate it. And it's how different are those withdrawal behaviors in the rat that gets that same opiate blocker injected that hadn't had sugar versus one that did have sugar. So the ones that did have sugar, getting the same dose of this opiate blocker, showed much more withdrawal. And one of the behaviors they do, they call them wet dog shakes. So that's one of the ways you can measure withdrawal is you actually measure, I don't know exactly how they quantify that, but wet dog shakes is what it's called. Just like a dog that's wet after they come out of the river or lake. The rats will do the shaking if they're in opiate withdrawal. So yes, there's models for that. Yeah. Any other questions?
I have one. So, obviously you studied the relationship between people smoking marijuana and wanting to get, whatever junk. And then there's the opposite side of people who have chemotherapy for cancer, and they get very nauseous.
Right.
So, is there a relationship between those two things, or are they different
Between
topics?
using marijuana to treat chemotherapy, or the side effects of chemotherapy,
and wanting to make you eat. Is that a different
and
topic?
wanting to make what?
And wanting to make you eat like junk.
Oh, right, yes.
Is that the,
Well,
are they related, or is that?
Yeah, that's interesting. So,
Repeat the question.
oh, just is there a relationship between the use of marijuana to treat side effects of chemotherapy, including nausea and reduced appetite? So, two side effects of chemotherapy: nausea, reduced appetite. The idea of is marijuana. Of course, marijuana is useful for both of those. Correct?
But what makes it reduce the nausea?
Yes, that's a good question. I could say I don't know, but
Does it just calm the body?
because I don't, but I can. So there's. Does anyone know? I need to ask that in case. No. Yeah, I mean, I would. I'm trying to. Opiates themselves, which acts in some ways similar to cannabinoids, they can cause nausea in the periphery. So there are opiate receptors in all over your body, but in the periphery they will cause nausea. If anyone's ever taken Vicodin or Percocet or OxyContin, one of the side effects can be nausea. So why would marijuana relieve that? That's a good question. I just basically gave you an argument why it wouldn't. So, yeah.
You don't know everything, man. I understand that.
I thought it did until today. Yeah. Right.
Did you have another question?
I don't know.
All right, if there are no more questions, I want to thank Matt for an actually engaging question and answer session. It was fantastic. Again, reminder, next time is volcanoes. That's right. I'm kidding. I'm making fun of it because it's her husband. And again, I want to thank everybody for coming out tonight. I appreciate it. And if you have any feedback of any kind, we used to have a number of mechanisms for feedback, but we do have a Facebook page which you can bludgeon with whatever comments you would like to bludgeon us with. We'd appreciate it, especially since we sort of flipped the environment in here extensively. And again, if we could just have a round of applause for Matt, that would be fantastic.